it's my pleasure to introduce Rebecca. She's been working with me for what, 4 months, 5, 6 months, maybe 4 months now, 5 months, something like that. Sometime last fall, she came to me and was interested in doing some research projects. And she works at she's a marine biology student, so she goes down to the sea lab. So one of the organisms that I work with is grass shrimp, and she was interested in starting some projects where when she goes down to the sea lab this summer, she could do some work. So this is some preliminary work, and those of you that were here earlier with Ansley Winter's talk on mine, using ImageJ, she's actually going to use ImageJ in sort of standard mechanism where we're actually doing measurements on these organisms. And she was trying to show that she could get these measurements and get them from the pictures to be able to show that they would work. So she was doing a lot of learning the program, learning how to work with the shrimp, learning how to culture them. So she did a lot of work on getting that done. So I'm really happy that she's got this book to where we present it today. And I'm going to let her talk about what we've been able to accomplish.
Okay. All right. So what we did was we worked with a specific grass shrimp. Genus and species was Polymontes pugio. So Polymontes pugio is a type of decapod crustacean that is very important in ecological food webs. They're located usually across like coastal salt marshes of the southeastern United States, Gulf of Mexico, and the Gulf Coast up to Maine. And Polymontes pugio can also be described as a transparent type of shrimp. It's small and very yellow, and as you can see over here, they're very transparent, very small, and they have like some brownish spots on them, and that's how you can tell that they're Polymontes pugio. And as detritivores, they are very critical in the nutrient cycling. They're also very important they're also a very important food source, and in many game fish larvae. They are also considered foragers and feed on a variety of items, including microalgae. Polymontes pugio also consists of 4 life stages, in which include embryo, larvae, juvenile, and adulthood. So they also have the nickname dagger blade because of their rostrum, which is right here, which you can see is very, very pointy. And female shrimps carry fertilized embryos underneath their abdomen, and until they are released into the water column and larvae is released, and that takes about 12 to 20 days. And the entire life cycle of P. pugio is in their natural habitat can be completed in as little as about 2.5 months. Some materials and methods that we did were that we had we got adult male and female shrimps collected at Dauphin Island, Alabama, at the Dauphin Island Sea Lab, which is down here. And we had them collected at Airport Marsh, which is located right over here on the poster. And when the shrimps were collected, they were brought back to a laboratory here at JSU. And before being placed into separate tanks, they were to be identified as either male, female, or juveniles. And length measurements were taken during this process. The shrimp were measured using a ruler, and if they were under 10 millimeters, they were considered a juvenile because we weren't able to see if they were male or female because they weren't fully mature yet. And if they were over 10 millimeters, that's when we were able to tell that they were male and females, and they were separated then. And the shrimp over 10 millimeters were inspected for their testes and ovaries and stuff. And the females were also recorded as gravid, and if not gravid, they were seen as how full their ovaries were on a scale of 0 to 3. And juveniles were placed into a 55-gallon tank for future use. Whenever they were grown, we were able to take them out and then use them as well for the research for future use as well. And the females were, the males and females then were placed into 3 separate tanks where we had 2 females per tank and 3 males per tank for every 3 tank that we had. And that's when we took record of it on November the 8th of 2009 of last year. And that's when we had the males and females. That's where it shows the length. And October the 18th when we collected all of them, and we had the length measurements for them over there, and when we had collected them, we had a total of 37 males and a total of 12 females and 37 juveniles that we had collected. And down here, we had the female, and as you can tell over here, which I said a while ago, the females carry their eggs under where you see the little darker shade is where she carries her egg underneath her abdomen. And over here is like a little more clear picture of what you can see. It's kind of just zoomed in, a little brighter, more easier to see kind of thing. And let me see. So the tanks for the males and females were fed daily and expected for gravid females. And 2 length measurements were taken on November the 19th, which shows right here on the graph. And they were all measured in millimeters as well. And in January the 2nd, 2 gravid females were observed, and the area measurements were taken, and a number of embryos were counted. And selected eggs were placed in a 27°C incubator, and length, width, and area measurements were taken of the embryos until they were fully hatched, which shows over here that they were kept in a 27°C incubator, and it shows that we had length, width, and area measured throughout time, started from day 6 and ending at day 14. And as you can tell, you actually see a growth throughout time, showing that that there is a difference throughout time, basically. So, yeah. And during this process, we used, as Dr. Rayburn said, we used Image J, or I used Image J, and I learned how to use it. And it was very, very useful. And Image J is basically an open source image processing program designed for scientific multi-dimensional images. And what we used Image J for was to determine the area of the egg mass, the embryo length, width, and area. Pictures were taken using a dissecting scope and an app called MultiConnect, and analyzed using the free Image J software. The eggs that were extracted from the female were all measured using Image J by setting a 1 millimeter measurement using a ruler placed in the picture. As you can see under here, this is, I basically set the scale to 1 millimeter measuring from one side to another, and I was able to measure from that the length and width and the area of the egg. And the data was then inserted into Excel. Both female ovaries were measured using Image J by measuring the area of the egg mass. And so by doing that shown here, as you can see the yellow outlining, that's what I did to measure the area of the female ovaries on there. So our original plan was basically to find a difference, like measuring the egg mass and probably getting data from. But our original plan didn't really work out. So instead, we decided to just work with the eggs and see their growth throughout time. And the results showed that in November we had no gravid females, but we did collect 2 data points for clutch size and area in January. Length measurements of the embryo over time were measured over time, and successful growth of adults in the laboratory also showed a good result. And as a conclusion, for future research, we will need more data points for area and number of eggs. Embryo measures may prove useful in estimating quality for embryos, and Image J was successfully used for this analysis. More data will need to be collected before field information for this research. So the significance of this study was to create a non-invasive way of collecting data out in the field and getting estimate of number of eggs being produced by females, by collecting them, taking a picture of them, measuring the area, and then releasing them back into their normal habitat. And it creates an easier way for researchers to collect data and not go back and leaving the species back to where it is. Instead, just taking a picture of it and then putting them back into their natural habitat without having a non-intrusive habitat for the species in particular. So future work will be with the collection of more data. There will be able to we will be able to have a more accurate data set. And therefore, by proceeding with this experiment, we will be able to see whether our experiment will or will not have a benefit to the environment, and hopefully make research a whole lot more easier in the future. So yeah, does anyone have any questions? Oh, one? Oh, just kidding. Okay. Who has a question?
All right, so why did you stop at day 14 on the embryos?
On the embryos, because that was when we started seeing shrimp, like little shrimp starting to form, and that was when we knew we had data results, like that was when we ended that, yeah.
They were hatching out.
Yeah, we had baby shrimps. Shrimp. Any more questions? Yes.
Is the fact that there were only 12 females and 37 males, is that a typical breakdown, you know?
Usually there's like, well, Dr. Hensley was the one that went to go get our shrimp for us down at Dolphin Island. And I feel the reason for less females probably because the season or like the temperature of the waters might have an effect to it, or really because when we had a whole bunch of males, and that's why we had like such little data, because it was hard to like get eggs, and so we were still like working towards that. Yeah. Any more questions?
I'm sorry. I was going to say, no more questions, but comment. Racing those eggs like that, she's much better at that than I am. That's not as easy as it looks trying to do the mouse to go around those guys.
Yeah. Okay.
Well, that does it for this little bit. We're going to have a short